Good morning. I'll be presenting on early detection of a phytopathogen, plant pathogen, which is Ralstonia solanacearum. This is in continuation to my PhD work, where I tried to explore a pre-symptomatic identification of Ralstonia in plants. So I will quickly go to the objective of work, followed by a little bit of literature review. I'll discuss the outcomes of my previous study and then highlight the importance of this work. So the objective is to develop a DNA-based biosensing platform for Ralstonia solanacearum. It's a notorious phytopathogen found in soils. It's a soil-borne bacteria. It is widely found in different regions, including India. The objective of this work is, can we have a pre-symptomatic identification, that is, even before these symptoms appear in plants, can we detect this phytopathogen in the plants or soil so that we can prevent the infection to spread. As we know, plant diseases can be caused by various factors. There are biotic as well as abiotic factors. What we are looking at is a pathogen. So it's a biotic factor which would involve to specifically detect. So a very important component of the recognition element of a biosensing platform for this type of application would be a recognition element like DNA antibodies, which would specifically bind to the target. Why do we have to detect plant pathogens? Because we know that 40% of the land is the agricultural cover, and pathogens in crops will not only reduce the yield but also the quality of the agricultural products. And highest losses are estimated due to plant pathogens all over the world. The traditional methods of how do we detect plant pathogens would be microscopic visualization, PCR, or ELISA. All these are traditional gold standard techniques, excellent, but they do have some limitations. Like you require skilled labor, you need sophisticated laboratories, you would require time because microbiological work would involve 2 to 3 days for identification. That's why, to overcome all the limitations of traditional methods, we have biosensing platforms which are used for various applications, and we can also use them for pathogen detection in plants. A biosensing platform has a recognition element, it has a transducer, and then finally it's the signal what you measure. So recognition elements can be of different kind. You can have an enzyme substrate, you can have DNA, you can have bacteriophages, you can have antigen antibody. I have chosen to work with a DNA for the stability it carries, especially thermal stability. Various transduction platforms can be exploited to detect this recognition phenomena which occurs. I would be talking about an electrochemical platform, which is an impedance-based spectroscopic measurement for the detection of DNA hybridization. So electrochemical impedance spectroscopy can be of two types, ferritic and non-ferritic. The advantages of using a non-ferritic approach is you're not going to use of any redox labels. It's kind of making it much easier, and you can perform your measurements in just buffer and try to capture the hybridization which occurs on the electrode surface. So this is the general scheme of my work. I have gold interdigitated electrodes which are fabricated. Basic characterization of the electrodes are done, and a DNA, a very specific DNA molecule, is immobilized on the gold electrode using thiol gold covalent bonding. Unwanted sites are blocked with mercaptohexanol, and my sample is dropped on the functionalized electrode. And if it has the corresponding DNA, if the hybridization takes place, the change in impedance is observed with and without the DNA sample. So this was the general scheme which I worked on to propose or develop this biosensing platform. Then I have applied it for various real-time applications. So DNA hybridization on the electrode surface, how does it work is, so when you have a gold interdigitated electrode and you have fixed your DNA, which is a single probe 20 nucleotide sequence, it's kind of like a layer which is formed on your electrode surface. As I mentioned, it's a non-ferritic approach, so you're doing all your measurements in PBS buffer. So you have a type of characteristic impedance curve which you will get. When you have this target sample which contains the corresponding DNA, and if the DNA hybridization takes place on the electrode surface, there is a change in the conformation of the DNA. This hybridization kind of creates or opens up the pores for your ions of the PBS to interact with the electrode. So you will see a kind of drop in impedance if hybridization takes place. So this kind of chemistry is studied and tried to estimate or quantify using impedance spectroscopy. So in my previous work, I have reported about zero point 0.1 nanogram per microliter of the DNA target to be detected using this proposed technology. And the device was quite stable, tried out various other cyclic voltammetry studies, stability, specificity. I also checked its cross-reactivity with other commonly found soil microbes, and this work has been already reported and it proved to be stable. So my next objective was, can this also identify before the symptoms actually appear on the plant? So that is my work on a pre-symptomatic identification. So how do we get about pre-symptomatic identification? So what I did is I took collaboration with Indian Council of Agricultural Research, ICAR in Old Goa, where I grew a number of plants in glasshouse as well as field and tried to study the life cycle of this Ralstonia solanacearum bacterium to understand when would the symptoms appear, when is it gets infections. I observed that it takes about 7 days for infection, and if you see day 1, day 4, day 7, samples were collected and they were tested using the gold standard methods which I mentioned earlier. The basic test would be just cutting out a stem and you see the ooze dripping from the stems of infected plants. All these, and if you see the images, day 0, day 1, you don't see any symptoms on the plants, but day 4, you start seeing the wilting, and day 7, it's a completely wilted plant. Soils and plant samples from all of these different stages of infection were collected, which are already tested with the gold standard methods just to make sure Ralstonia infection is present in the plants. And these samples were tested on the proposed biosensor to see when, on especially on day 1, when there are no symptoms in plants, can the sensor detect the presence of Ralstonia in the soil and in the stem of plants? So I observed a tremendous change in impedance of with the samples which were collected from day 1, day 4, and day 7. So with this observation, I can conclude that the proposed biosensor is able to detect the target Ralstonia solanacearum in different parts of plant even before the symptoms appeared on plants. So that's my conclusion of the work, that the proposed sensor is able to detect Ralstonia solanacearum infection even before the onset of plants. So it proves or revolutionizes to have an early detection so that appropriate measures could be taken to prevent the disease spreading. Thank you.